Okay. So good afternoon, or good morning, or good evening to everyone, wherever you are joining us from. My name is Lamarana Jallo, a PhD student in biochemistry and the American Society for Microbiology Young Ambassador to the Gambia. It's my great pleasure to welcome you all to this inspiring episode of the ASM Young Ambassadors webinar series, which is proudly brought to you by the American Society for Microbiology. This session, as has been stated in the previous one, is co-hosted by myself, the Young Ambassador to the Gambia, and Dr. Leisure, who is the ASM Young Ambassador to China, in partnership with supporting institutions, namely the Antimicrobial Resistance Research Group at the West African Center for Cell Biology of Infectious Pathogens, that's WACPI, in the University of Ghana here, which is led by Dr. Abiola and the Gambia Society for Microbiology. The purpose of these webinars is to foster scientific exchange and collaboration, connecting students, early career researchers, faculty members with global leaders and innovators in microbiology and related disciplines. Our goal here is to strengthen the microbial research and education in the Gambia, across West Africa, across Africa, and globally for a healthier and a more sustainable planet. And today, we are honored to host an exceptional scholar and leader in the field of genomics and public health, who is Dr. Abdul Karim Sesay. So a brief synopsis: Dr. Sesay is an assistant professor and the head of the Genomics Strategic Core Platform at the MRC unit, the Medical Research Council unit, the Gambia, at the London School of Hygiene and Tropical Medicine. Since joining the unit in 2017, he has transformed its genomic capacity, establishing one of Africa's most advanced genomic laboratories and leading critical efforts such as the COVID-19 sequencing response in West Africa. Before his current role, Dr. Sesay served as the assistant lead of the Advanced Sequencing Facility at the Francis Crick Institute in London, in the United Kingdom, and also previously served as the head of the Genomics Facility at the Medical Research Council, NIMR, where he introduced cutting-edge technologies such as the Oxford Nanopore sequencing. In 2022, Dr. Sesay was awarded a Member of the Order of the British Empire, that's an award by Her Majesty, the Queen of England, in recognition of his outstanding contributions to genomics and public health in both the UK and the Gambia. Dr. Sesay has an impressive academic record with over 100 peer-reviewed publications, reflecting his scientific contributions to the field of genomics and public health. And today, Dr. Sesay would be speaking on the topic Advancing Pathogen Genomics in Africa, Challenges and Opportunities, a subject that sits at the intersection of scientific innovation, health equity, and continental development. So the format of the webinar is going to be 40 minutes presentation and 20 minutes question and answers. So please note down any questions or comments or suggestions that you could have in the Q&A section. They will be related to Professor Karim once he is done with the presentation phase. And please join me in warmly welcoming Dr. Abdul Karim Sesay to this session. Prof. Sesay, the platform is all yours. Thank you.
Okay, thank you so much. I don't actually recognize myself with that kind of introduction. Really, one of the things that is so exciting about doing something like this is that we're connecting the dots. The globe is really very, very small. Actually, I mean, it's so small that a couple of days ago, I was in Ghana and I met Lamarana in Ghana. So we both Gambians in some way, mine a convoluted Gambian, but we met in Ghana because I was there for a meeting, and he's been an aspiring young person, and it really is a pleasure to be able to do this. And hopefully, I like stories, and I think if you really want to tell somebody about what you're trying to do. It has to be like a storytelling. I think that that's what I'm going to try to do. Hopefully, it will be an interesting story, and there might be something you will take from that story that might inspire you and show that yes, we need technology in our region, and yes, there's something we could do, and yes, there is so much more that we need everybody to collaborate to move forward. So my thoughts is that whether I will cover that as the title for my presentation will go, but hopefully some of it you will get, and I'm hopefully again I'll be able to keep to time. I can talk on 40 minutes, although it looks long, it might not be too long, but for some of you it might be far too long, but some of you might be short. All right, so yes, I've come back to, as Lamaran has pointed out, I'm not going to come back. So my journey into Africa, establishing the genomic facility, the huge thing about the pandemic means that I don't have to explain to everybody what genomics mean, and capacity building and training, which is the reason why I came back, and really the core platform as a cross-cutting technology platform within the unit and hopefully within the sub-region. Okay. It's a slide that I think all of you, even though most of you might be microbiologists, that should be familiar that the capacity to investigate genome is a huge thing. But really what's important that's in the red is there's a real huge interest in implementing genomics in Africa. The reason is that quite a lot of samples are collected in Africa. But the big thing is that most of that work is not done in Africa. So there's a really, really big push to make sure where samples are collected, that's equitable for the samples to be processed, and also whatever benefits and whatever impact that will reflect in terms of what you want to do in your country, not that. And COVID has showed that it's really important that you have technologies within reach for you to be able to respond in context to whatever a situation that you dealt with. Okay. Yeah, state of play in Africa. This was 2023. I think there was a meeting in Uganda, and we were all complaining about the fact that samples are still sent out in huge numbers, and there's lots of reason why people send sample out. And what I'm not going to try to do is to do a negative conversation. What I'm going to do is to say, actually, all of the problem gives us opportunities, and the opportunities is what I want us to really discuss and exploit anyway. And yes, we have a resource of samples that can be collected, but also because of the diversity, also because we are burdened with diseases, then there is a really huge reason for us to be able to sequence and be able to do some of the cutting edge technology within reach, because that's the way you will train people. That's the way you can actually develop and actually do innovative work, because you have that power within your hand. You have ownership. Okay. Opportunities. Are you able to manage genomic technology to be able to show that within our context is such an important thing, to get resources in, resources that are science-driven, that you can establish protocols that you could use within our setting. We have lots of challenges, and those challenges has to be contextual, electricity, power, water for some lab, getting logistic to bring samples in, custom, cold chain, and all of those things. But those are opportunities that we could do. How do we organize training so that our continent is really it's large, but we're quite interconnected. I can get to Senegal, I can get to Sierra Leone in 2 days by road. I don't have to and our people are also moving the same way. So really setting up all of those things within our setting, it's a really huge opportunity for us. Okay. Yeah, so for me, and this slide shows that to have a state-of-the-art genomics core was just a no-brainer. And that's the reason why I'm back here, and to strive to improve and evolve current, and also not just be copy and paste, but really do protocols that are contextual to our region is really very, very important. But we don't work alone. How do we engage stakeholders? And then stakeholders comes in all different guises, from our population, politicians, policymakers, all of those things. Those are all important. And if we're talking about genomics, it's not about human genomics. What about animal genomics? What about environmental genomics? All of those are all of the aspects that you have to put together to have really something that you can manage that could make a difference to our population. So hopefully you have some flavor of some of the stuff we've been trying to do. Okay. I like this slide in some ways, that when people think about technology, you get the best machine going. So I use an analogy like you can buy an iPhone 16, but in fact, if you don't have a young brain like your guys that can actually operate an iPhone 15, for me, probably you can just still give me an iPhone 8 because I don't have all the skill set to be able to operate that. So I start first about individual level. I'm the training that I've had, the collective skills that I've maintained with the time I've spent out and time I've spent inside, to be in a structure, whether you end up in the right lab to be able to do the right thing, and they give you the opportunity to be able to flourish. It's also very important. And are you involved in not just your research, but are you involved with the government? So Alamara just pointed out that one of the things I'm proud of is that part that I also involve outside of the research and do. And again, you don't stop there. If you got technology and only you is doing it and other people are not doing it, you don't make the impact that you do. So there should be a capacity, the reason for you to capacity develop your neighbors and collaborate with them. And then hopefully with all of that, you might be able to contribute to something that is worthwhile. So there's 3 buildings here. I started in a very old building in London that got moved to a very fresh building, also in London, but more central. And then I'm now, and you can see the last building is Gambia and a new molecular lab. And it's the echo surrounding the unit is really beautiful. It's green and it's close to the sea. So the sea is just at the end of those grassland. So it's really a really beautiful setting. So I can walk to the beach in a couple of minutes. Okay. So again, with technologies that it's not, you need to have an introduction. So I've been introduced to Oxford Nanopore technology from when it's inserted. I started on genomics in 2010. 3-4 years later, Oxford Nanopore released their platform. I had a project and I asked whether I could use the platform, and I've built up a relationship with that, and that's paying off. I mean, at the end, you will see what I mean by it pays off for having this long-term relationship. So this relationship is 11 years. So there's a picture of me and my boss. He's been very, very proud that I am one of the 600 ONT platform in the world that was distributed for project that they supported. Okay. The unit as the core, which I am now, is kind of a triangle with the facility. That's the genomic facility that I established when I moved to the unit. The data, so we have a HPC for the bioinformatics and the data storage and data analysis. And it fits in really well with the school. So we're part of London School, and quite a lot of the mission is about health, research, education, and how do you translate those knowledge into policies and practice. So I'm hoping that during the talk, some of those things we've been able to achieve. Okay. I came in the unit 2017, but the unit's been here since for 78 years. So some of the things is not, I can't claim it. I mean, I could claim what we've done with the genomic facility, but there's a really good biobank in the unit. There is other facility infrastructure for computational was already been thought of and established. And also they saw a gap that it needed genomics, but the molecular thing, there was infrastructure and a core area for the molecular. So it really fit in really well within that structure. And then I started bringing equipment. This is the map of the Gambia. apart from the sea on this corner of the map, all of this is Senegal. So Senegal surrounds Gambia completely. So it's a river is named after the river, and this why it's the Gambia. But I'm using as a backdrop to the platform. So we've been able to science-driven added different technological platform in the unit from when I got here in 2017, going now to 23, 24, having and over 6,000 in the unit. So some of them have been very, very successful because of the use they have, and some of them we're still thinking of how to utilize in the best way possible. Okay. For me, this is kind of a nice route about where I came in. And really, one of the most important parts is that during COVID, I was seconded to the Minister of Health, the National Public Health Lab. And again, it was a really inspiring time for me because they did not have a molecular lab. And I thought that they could also have a molecular lab. And it was really a very successful partnership because over 80% of all of the all of the testing for COVID was done by that lab that we set up and generate a lot of fund for the country and for all the travelers that need COVID testing. So this is really one of, I think, the best thing that you do. So we're looking now to 2025, probably in the next couple of weeks, one of the platform will actually designate us as a center of excellence for their platform, which is actually a big thing because it will be the first one in Africa and probably the second one for that platform globally. Okay. In terms of the unit is divided into themes, and I've been fortunate that I have collaboration with most of the people and most of the themes within the unit, either as support for them, co-investigator or co-principal investigator or co-collaborators. So it's been working, and quite a lot of my staff are funded. So most of the units and staff, apart from what I call the core people, have to be funded by external funding, competitive external funding. And we're still in a good position where we are still applying for grants and within reason have a reasonable success to be able to keep a team together to do some of the work that I would present. Okay, we're doing time. We good? Okay, yeah, so, and again, this is a flavor of some of the projects, and the red are some of our collaborators within the unit, and I don't want you to chase the slides, but there is a huge difference in terms, if you look at the black circles, we're doing bacterial and viral metagenomic, single-cell transcriptomics, bacterial fungal metagenomic, WGS, AMR, really working a lot with pathogen and really all of our work around pathogen genomics. But we're also doing human stuff as well. So that we're using human genome work where we're looking not just at pathogens, but we're also looking at host. And again, we all have a project where we're looking at vectors as well. So it's not just pathogen. I think having that link between pathogen, environmental, and other things is really very, very important. Okay. There was a West African platform in the unit, even way before I came in, way before COVID. So COVID wasn't why we set up the network that we have. We had few network that the units were involved. Some of them followed with me. So for the last one, for example, one item and a climb, I'm really actively involved with that. But some of them were not mine before I came here. But most of this now, either my team is involved in delivering training, been involved in and some work packages within those networks. So it's really, the unit is really well placed within the West African region. And I would shift, then we get to COVID. At the first COVID case in Gambia in March, and we had that sample. Actually, the first COVID case in Gambia, we sequenced that person samples many times because there was this thing that COVID will evolve so fast that even within 1 or 2 weeks, there will be new mutation and so on. Of course, that it didn't pan out that way, but everybody in the world were over sequencing. But we had the facility, and so we had the sample within a couple of days of the first COVID. We would sequence it, and we followed that. That's all during the pandemic. Every single COVID sample in Gambia that were positive and that have the CT values in the right way, we would sequence it. And that was really very, very successful. Again, because we had this very, very, very strong partnership with the government. So they collect samples, they test samples, and then those positive samples, we get it in the lab, and then we sequence. And my student, Abdullah, does all the bioinformatics, and he's done really good work in showing where what variants we have in the country when it was introduced, and how that is how that spreads out within the country, and how that matches with what happens globally. So these are also phylogenetic through trees, which shows that I'm looking at all of the sequences for the 200 bit years of COVID different wave, and where how do they group, and where did they come from, and how they are transmitted across the world. And again, this is a phylogenetic frequency over time, looking at the different variants and how does that look like with our data compared to global data set. And it really shows a really good confidence that the samples that we were collecting, we were sequencing at a really good time as well. So we were not doing retrospective sampling. We were doing real-time sampling of COVID as they introduced in the country and been done. So we were doing really quite well. And I put some timeline on some headlines. So in September 2020, I said that everybody can do their own sequencing. If anybody invests 20K in that country, they should be able to do their own sequencing. And I think sometime in March 2021, so a year now, then there was this article, I think in Washington Post, that was trying to embarrass the American government about they are not sequencing enough of their positive. And they would say, oh, Gambia, this small country that you can't even see, they managed to sequence a high percentage of the positive. But this is a number game. Gambia did not have hundreds of thousands of positive. Our numbers were reasonable, but still, what it meant that we were really doing very, very well in terms of sequencing. But what was sad is, and you're not going to see this in the last table, is that the countries around us, even at that time, really a long time after COVID, they were not able to sequence their own positive. There were people who were supposed to send their sample. There were issues in moving stuff from one side of Africa to the other. So people were not able to sequence the samples. So I thought, why not? I mean, we could do it. We are able to do it. I managed to do something in Gambia. Why not try to do other countries in the region? And in the same time, WHO and also WAHU thought that, oh, these guys had the capacity, so why don't we don't designate them as kind of a center of excellence to support the other countries. So I took it on myself with my team members, and with the dots, we managed to visit those countries and establish sequencing facility for them. So it's not, so they don't send us samples, so they sequence their own sample in situ. And so from bigger countries like Nigeria to smaller neighbors like Guinea-Bissau and so on. And it really worked well. And we trained people locally within a couple of weeks, and they were able to sequence their own sample. And we had this structure. It's really something that I really like, that I developed within my team, to say, actually training as this aspect of, it needs to be structured. Otherwise, I mean, what happened in our region, people get trained. Sometimes even the bosses get trained on working in the lab. They're never going to be in the lab, but when there's a training, they go, because it's not structured. It's not, nobody follows the training after the training. So whether are you follow it, are you coaching the people, are you mentoring them, are you monitoring and evaluating, are you seeing whether they've learned anything, and are they sustaining their learning aspect to do? That's a lot of investment to put that structure in place. And so, but I think this is the best way to actually get a good, clear view whether the training that you are delivering has an impact. Because if it doesn't have an impact, all you're doing is just repeat it, repeat, 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 and nothing is achieved from that. So yeah. Again, this is another slide that elaborates some of the training aspects, but it's not in silo. I mean, one of the things that I really like about my background is that I have a dependency on the South for some of the support, even financial support to be able to continue this training as I am with my neighbors in the North. So that's really very, very important that you're working with partners. Some of them have skills that you don't have. Some of them can provide resources that you don't have. And at the end of the day, is delivering a more effective training regime and really making collaboration that moves things forward. So it's not, but it's very difficult because when you look at it, is that partnerships are formed with interaction. And some of our younger people cannot travel, they cannot get visa to even make interaction. So how do they get access to these kind of things to do? That's something that, we should be working on. How do you expand your network to include younger people to also join that network is something that I think is something that's exciting for me, is something that we should be working on. I will go through a couple of projects. This is one where we're trying to sequence whole human genome, getting methylation and also genomic information. And one of a young lady in the lab has been doing really well. And one of the things I like about the slide is that the manufacturer will tell you can get so much out of our machine, but in our setting, can you do that? And one of the things Mariam showed is that whatever it says on the tin, she gets what she says on the tin. Yes, with a lot of tweaking, with a lot of help, really a lot of help. They send someone around to support us to make sure that we get the protocol and it works. But what it does say is that you can do it within our region. If they said you get 150 gigabytes from one flow cells, we get it. Mariama gets it, not me. And that's really very important. And it shows quality of data from when we started with a lot of twig. The one that's the green is the important bit. You can see it was dying down quite quickly. But with lots of tweak and lots of work, you can get this to get more data. So green means more data. Okay. Another project where we wanted to generate really high quality plasmodium. So most of the time, if you don't have a good reference data, then it means that what you're calling as SNP and SNVs and indel might just be from the reference, but not from contextual from what you're doing. So what we wanted was to have field plasmodium genome at high and quality, high coverage, to be able to use that as a reference so that you're comparing apple with apple, not apple with pear. Might look like an apple, but it's not an apple. So this is really important, generating. This is something that Mariam, MSc student, now gone to do a PhD, was doing, and shows that we can get very, very high coverage, which shows the bioinformatic tools, and those slides show you very, very high coverage of a plasmodium from the field, not something that's just been grown for the last 100 years. These are field plasmodium, and we could generate very, very high that could be used as reference for new mutation and follow mutation round. So this is just elaborate, and you could see for all of the different chromosome, you get very, very high coverage. Okay. And she wants to do more, and this is not her, but a boss, Nora, wants to do a little bit more with what we can do with the sequencing. Same platform again, ONT platform, and actually looking at diversity. And so you can follow how the parasites is shifting across the different region in the region. Okay. So that's one on the plasmodium. This is a project I really do like. And again, it's shifting not from human stuff or the traditional stuff that we've been doing in the unit. So this is a vulture project. And actually the project started in more as a curiosity. So one of the things that the platform was doing was to say, if you can sequence, find a clinically endangered species, whether it's plant, animal, or anything, they will support you for you to, if you got the platform, for you to be able to get genomics data from that species. And Gambia has 4 different types of vultures, and 3 of them are on the endangered, critical endangered list. So the first bit of the project is to get really good genomic information from vulture. Vulture is really important in our region. They are kind of the scavenger. And there is an inverse relationship that if you kill all the vultures, then you increase vermin population in the country. So really they are, and they also become a reservoir because they eat dead animals. So if animals that have been infected with, and again, like dead bird flu is passed on to the vulture. So we wanted to understand not just the genomics, but also what is the reason why we are losing vultures so critically the endangered list. So we're looking at what the metagenomics, looking at what they eat, what they don't eat, and what they interact with, and what pathogens they're carrying. So it's a really nice project, and I've got sharing with somebody who's working on mostly, I would say, on ones who is interested in environmental factors and really the link between environment and the changing environment and the animal that are residing in our country. Okay. Another thing that we did, the slide doesn't matter, but the actually idea is that we do collect a lot of sample. And what is important is, are those samples fit for purpose? So what you can do is that if you decide what you're going to use the sample for, whether it's DNA, RNA, you at the time of collection and different type of collection, you extract those RNA, DNA, look at the quality, and then every year you can assess whether the freezers have kept the temperature, nothing has gone wrong before you start saying we got valuable sample, you're paying lots of electricity. So we started this idea that biobank sample of big projects, we will quality control it, do a few quality control metrics and make sure we keep that, and then we can repeat it as time goes on. Okay. For the same guy, he's doing another project where we're actually doing multi-omics. So it's not just genomics, but interested in metabolomics, epigenetics, transcriptomics, and also proteomics. And that gives you, if you think of a puzzle, genomic is just only one part of the puzzle. I think that if you can get all of the puzzle pieces together, even for pathogen, looking at the readout is metabolomics. If you can get a metabolomics readout, then it's really much more effective. So AMR and AMR, how your body reacts to AMR, or how the bacteria process the antibiotics to be resistant, as a readout as metabolome. So you could do that. And it's been done with mass specs that people are using it for working out AMR, for working out the different bacteria. So there is a signature, and you could access that not just on genomics, but transcriptomics, proteomics, or metabolomics. Yeah, so a project that has quite a few samples from Kenya, Mozambique, and Gambia, and we're going to do all of these things to those samples. This moving from what I call human, animal, or the other project we'll be doing, this was an extension of COVID. And the idea is that, could you see COVID in wastewater? It's not intrusive, and you don't have to label someone, but you can imagine that the way composite sewer sample are collected, you can follow infection. So you can follow infection maybe in a sewer line out of an hospital and see if you can actually, if you cannot tell as early detection, but you could tell whether, in fact, a particular pathogen's been circulating in the hospital. And really, if you can link that to outbreaks, mini outbreaks, that hospital-based outbreak, that would be a fantastic readout. So we started as a pilot and looking at wastewater, a couple of sites in Gambia, and basically have samples in 1 year from 4 sites, sampling and then looking at different viruses. But also we took those samples and also grow bacteria from it. So this is a process, the method for doing that. But make sure we concentrate virus. You do the same for bacteria if you want, with different kits that you can do. Then you do quantitative PCR, and from those you can sequence and look at bacteria classification, or even COVID sequencing that are amplicon-based and COVID sequencing. And really what we say is that we can see COVID from the samples. About 43 of our 180 samples were positive for COVID. Really, at this time, we're talking about the Gambia, because they were not doing travelers, we're not reporting COVID positive. So we know that COVID is in the country, it hasn't gone anywhere, and we're still in circulation. This is at sewer level. So, and there are other viruses that we also can detect from the samples that we collected. And similarly, we started also looking at bacteria that you can got from those samples, do bacteria culture, and then look at those bacteria. So we started that again in collaboration with the National Public Health Lab. And the Gambia has really sporadic Klebsiella pneumonia outbreak, which is one of the biggest kind of as a killer for young, very young people, infants and neonates, are sepsis that's caused by Klebsiella. And what would be really nice is whether you could see the Klebsiella that we see in the sewer sample before, if we can get whole genome data and see when there is an outbreak, can you see its early detection to know, okay, so you have a problem in the hospital, and that you could do preventive measure, or you'd be a way to assess whether you're clearing the infection at the hospital level. So something with more intense sampling we'll be able to show. Okay. Again, there's another more on viruses and cancer and HPV, what is called hepatitis B virus. In Gambia is a problem because you see young men that have not been immunized with liver cancer that's caused by hepatitis B virus. So what we want to do is to look at the genome of some cohort from Nigeria, from a RF fellow, and a cohort from a project that's been going on in the unit for a long time. So this is ongoing, and we're looking at the genome and try to have a better understanding of the different genomes, hepatitis genome in the country, and whether that will be a link between severity, what they call drug resistance going forward. So these are some of the stuff we want to do. All of what I say is that there's things that come in my mind, whether can we do equitable research in Africa? Usually people will say probably not. It's not a reality, because and the difficulty is that what you get, even with our collaborators, is that what you do get is what they call equality. It means that they make the size is still the same, but it doesn't fit our purpose. What we really do want is kind of a liberation. So seems quite what's it called, quite shake over in terms, but really you can do this. I mean, if you take this word map and use the word, if you can do it with the people trust and respect what you're trying to do, which I think is what I got with the Ministry of Health. And also I'm working, also it's not at that level. You can go back to educational level. And one of the things, how I met Lamarana and one of her friends, is that I've been trying to work with the educational part of it. So working with the university, trying to get them to be able to have access to cutting edge technology just for teaching, so that people have been taught not just by a textbook, but actually by using a pipette and using micro pipette and being able to do manipulations, being able to run a gel, being able to do some of this molecular technique. If you really want people that trained for the modern world, you need to train them in modern technique. So this is also something that I feel is really very, very important. We publish. I've got lots of people that I thank for what I do. My family in the Gambia, they are family now, although I wasn't born in Gambia. My folks are Gambian, and I've been quite happy to come back and learn a lot about my family. So with that, and some of this you might get it, but if you are not English and you don't get this particular poster, then you can ask me a question. But otherwise, thank you so much for listening to me, and I'm quite happy and open for any question from you guys. Thank you.
Thank you so much, Professor Karim. This has truly been an inspiring and an enlightening presentation. Few things stood out for me. which is you mentioning that you are in the verge of having one of your labs or your centers being a center of excellence, actually in like being one of the first in Africa and even perhaps second in the world. This is very impressive and demonstrate that scientists in Africa can also take the lead in pushing the boundaries of science, not just following. Another thing that really stood out in your presentation for me was the part of your presentation where you highlighted that over 90% of sequences of African samples is still done outside the continent. Really stood out. This really stood out and it lingered in my mind. This underscored the urgent need for Africa to take ownership of its genomic data and infrastructure. It shows the power of genomics not just as a scientific tool, but as a driver of health sovereignty, innovation, and equity on the continent. And their journey and leadership are proof that with vision and commitment, Africa can also lead in this space as well. So I'm just going to open the floor for questions and answers. I'm sure we are going to have many people who want to ask questions. And please post any questions in the Q&A section. I would forward them to Dr. Karim to answer.
Thank you so much. It's a pleasure, and it went so fast. I thought I only done 5 minutes, but it's really good.
Sure, sure. It has been a very wonderful presentation. The questions have already started flowing. Somebody asked, is joining the Genomics Hub open to us? I am not sure from where the person is talking from. But yes, that is the question.
Oh, okay. I think one of the things that is really very important about when you talk about hubs, nodes, or whatever, I used to actually work in neuroscience. So, and I actually feel the analogy about your neurons in your brain is really very important. So, connections, you can use it. If you don't use your connection, you lose that connection. So one of the most important thing is that if you have something that aligns with somebody and that's quite active, then that person is your partner and you can work with it. But I think some of the time we make partnership and it's actually not linked to either your interest, their interest. Like your neurons, you will lose it. So I think so I'm open. I'm only one person. I'm hoping that it's a connection. So because you know me, I know somebody else. It doesn't necessarily mean me that will have the link. It's just the connection. So it's open. I mean, I said one of the things I really like is the interaction with younger people because we have only a finite time, a very short time in terms of what we will do. But younger people have much more time than I have.
Okay, excellent. I believe that have answered the question. So the next question we have here is how reliable are genomic data outputs from mobile or field deployable sequencing labs in Africa? And this individual asked two questions. I'm just going to push them all so we can skip another one. The second one is how can African countries build sustainable genomic surveillance system beyond donor funding?
It's a fantastic question. So I will start with sustainability. Sustainability is some of the thing that really worries me. So what we do, especially at public health level, and the reason at public health level because there's not many African countries where the government fund science or research or surveillance. There's not many. So if you go around and really, one of the eye-opener at the moment is the political situation we have with America, where people funding are being pulled. So whatever HIV, TB surveillance were a national program, they are not funded by our countries. So the answer is that we need to have open partnerships. So if you are working and you're working with a research institution that are very good at getting grants, they're very good at getting ideas. So you might have young people, educational system where you are teaching, so you make some fun. So we need to be flexible. I mean, science and surveillance or whatever, because it's so important. We have to have a flexible system that doesn't depend on one system alone. Donor system, it doesn't necessarily mean that it relates to the country's need. And I think that has to be very, very importantly stated, that just because somebody's giving you money doesn't mean that he wants you to do exactly what you want to do. So that might be a caveat to said about ownership, about do. But sustainable stuff, whether you've thought about it, you have your plan A, plan B, plan C is the answer. In terms of technology, I think that when COVID started, there was a lot of noise about whether nanopore with the idea of the error rate, would they be able to call variants for viruses? And I think sometimes what we do in science is that we have a copy mentality in terms of saying, okay, so if you have a 98% error rate and something is 99%, so something that's lower might be a problem. But actually say it's you, if you have a think properly, is that if you are reading a mistake that you write, but you write it 100 times, if you count down the line, you will still have, if you counted the percentage, you will still have the correct line. It's a small virus. If you sequence it enough, the error doesn't matter so much because you've got such a high coverage that you're making more correct call, that the couple of wrong calls doesn't matter. But people didn't get it. I mean, and there was a big thing at the beginning. I said, no, it works and it will work really well. Other people knew this, it will work. But it's really funny, we're talking about having Chinese partner. The moment that the Chinese bought a couple of MinION, everybody said, actually, if the Chinese can buy MinION, it must mean it must be doing the right thing. So again, this is the copy-paste society that you need somebody else to say, yes, it's the right thing, and then you go and do it.
Okay, thank you so much for that elaborate answer. The next question we have here is what amount of training is required to be able to do meaningful work or research in genomics?
One other thing I say every time, every single time, is that I can train anybody who has a focus and an intensity to want to do it, anyone. So I think that's one of the things that is open to me. I've actually shifted my career from being a biochemist, being a I'll call it a neurosurgeon, but mostly doing neurosurgery on mice at a really high level, and then coming to genomics. But I actually have what I call energy level. And this is what I look for people. I really don't really worry about whether they came from Oxford, they have a first-class degree, that they're enthusiastic, they have the flexibility, they have the focus. And if they have all of those, they can be trained to become that. It depends really. One size doesn't fit all. So if somebody was going to say, I'm going to just learn wet work, that means working in the lab. That's quite different in somebody who said, I want to be a bioinformatics and be able to analyze my own data. So, but they might have computational background. So switching that to bioinformatics might not be easy. They might have molecular background. So switching that to genomics might be very easy. So I think said that your character is what would determine how long it would take you to do it.
Okay, excellent. We have one here, but this one is actually trying to express his gratitude. He said, "Thank you, Uncle Abdul. My share, I think Sekou here. This is Sekou, and he's expressing his happiness with the presentation." So
Thank
I
you.
actually had a question I was going to ask, but you've somehow answered part of it. It's coming. That was like, you've mentored a lot of young scientists. So what practical advice would you have for students in Africa or even early career faculty who want to get into genomics but do not have access to advanced lab equipment. And just going to the second thing I
So, yeah.
wanted to
No, no.
know.
And again, it's a really good question to have the right lab, the right environment, and for all of those things. It's not it's challenging because we are not. If you go around and you want to do internship, for example, the number of labs that you can go to is quite limited. I always say is that when you have a flexibility in terms of when you have to access something, then you have a chance because it doesn't depend on a fixed timeline. So, for example, if you say, okay, so I don't have a genomics training, I don't have bioinformatics, so I'm going to go do some online courses first and see how that goes, how I learned that, I practice and I do this. And maybe I would apply for a small thing to visit a lab or that kind of thing. But I would say the flexibility that you bring in into your career that helps you to said maybe it will be take you longer, but you will not stop you from trying to get to the right lab, get to the right people, and get it to the right individual that will train you. So I'm looking at you. I met you in Ghana. You had lots of good advices. You were patient, and all of those things comes into it. And we are very difficult to be patient because for we all TikTok what I call we are now TikTok society. So everything has to be now, then, and then it's finished. But sometimes it might take longer than that.
Okay, excellent. The questions are still coming. I'm going to check this one. It said, can we have the presenter's contact for the interest of training? Should I am Comfort Danchel Vandu, a PhD student in microbiology, and his research is in genomics. Sure.
I love it. I mean, one of the things that in fact, it's the yesterday, Lamarana reminded me that I'm going to give a talk. So I was in UK. I met him in Ghana, so it's really weird. So then I went back to UK and then came back to Gambia. So I lost 3 nights of sleep. And then this boy is telling me, this young man is telling me I need to prepare for a lecture. And so there is, I really have no issues in Lamarana sharing things. You be patient, also be patient with me. I might respond in the right way. But let me say one advice I give every single person that I deal. I said that one of the way that you can get the best out of contacting somebody is not say, if you send me a mail and said, I don't know what I want to do, can you advise me? That I'm never going to be able to advise you. Please, let's be honest about it. But if you come and said, you know what, I've got two cool options to do A, B, and C, and I'm trying to think about this. These are the advantage for these are disadvantage. What do you think? It's a very simple. I can just say, just choose that one and get on with your life. So there is a way that you can actually get people engaged in supporting you, but try to minimize and get the most effective answer in one sentence, not in a book, not in a thing. So, and that's important. So if I was asking somebody that mentoring, I still get mentored. I try to make sure that I really specify really what do I want from this person. And that will be, even though they don't have time, they will be able to answer this with very little time investment. So that's what I say. So I have no issues, but if you send me and all asking me for career advice, you probably will not get answers from me that might be useful for you.
Okay, that I believe that is well heard of too by the person that asked the question. I'm going to share my contact, and you can reach out to me. Then I would mediate your
That's
conversation
fantastic. I
with.
like
Yeah,
that. I like,
yeah.
and now I've got a nice filter in my younger siblings. Fantastic. I like
Ex-
that.
Exactly. So I'm going to share my contact in the Q&A in the chat box, and you can contact me right there. So
Okay.
we have another question. The session has been very interesting and interactive. So this one says, what key qualities, skills, or attributes are essential for someone to successfully pursue a career in the field of genomics, particularly in the context of research data analysis, and public health application.
Okay. You know, one of the things that is important for you as all of us, you know, one of the things that is important is that you have a big picture for yourself. So I said that you have the accidental picture, but you have a big picture. So when I was looking at myself and say, what do I want to do? What am I good at? And the first thing comes is that what am I good at? You know, what if I am a good number cruncher or somebody. Actually, I know I'm a good number cruncher. So that's already tells me what I can do. If you put me in a business to negotiate, that kind of thing, I'm going to fail because I think I don't have within my kind of the way I think that kind of mental state to do that. So I think what is important is that you recognize what your strengths are. So you work it out, you know, even the way you interact with your siblings, you interact with people, you work out the things that seems relatively easy for you. And then you can now make a decision about, because you want to do something that you will enjoy. I mean, it's so important that you enjoy what you do, and it seems quite easy. Actually, talking to you guys is the funnest thing I do. I really do have, I mean, I wish it was live and I can see you guys and I can have this really warm feeling about what you think or what you don't think. But yeah, we don't have that. I can't go around the world. But I think you need to enjoy what you do. And then it becomes easier to decide that's the skill that you need to move forward. Because if you don't enjoy it and you hate it, I swear you will, even if you're successful, it won't be fun. Do you get it? So
Yes.
let's
Sorry, comment.
yeah, no, it's done. It's fine. Yeah.
Okay, thank you so much for that. I just wanted to say, the other time we did invited you at the University of the Gambia, where you helped organize a very successful workshop on bioinformatics and then genomics thing. We would probably like to invite you again to many of our students in the University of the Gambia. Some of them wanted to attend, but they are on classes, others are engaged in some other activities. They
It's no
couldn't
problem at all.
attend.
No problem.
Sure.
No
No
problem.
problem. We'll do that. We are nearing the end of the webinar, but I just have a question I want to pose for those that unable to join. We are going to post this in our YouTube channel. The question is, can you share some of the biggest challenges you face in setting up an advanced genomic facility in a low resource setting?
Yeah. You know, one of the things that I need to recognize and identify is that the unit, the way it's set up is that getting equipment and the structure. So one of the things that we're talking about, the center of excellence, and I wanted to touch on this is that, so I really did show you deliberately a slide of having a connection with a platform from 2014. Okay, so that's a long-term relationship. So even if it's a girlfriend, that's a really long-term girlfriend, that's a wife. So that's very, very important that you establish a relationship that is long-term. So even where I'm now in Africa, for example, I can put this really and very proudly said for the ONT platform, one of the only African institution that can still order stuff from them directly without going through a distributor. And the reason is it's a long-term relationship. As I said, that's really very important that you form very, very good partnership so that even the challenges that other people are facing, like, you know, cold chain or the all of the kind of thing, those are all something that kind of is in place, you know, not perfectly. I mean, we are okay, but we are still in Africa. Things right now, I'm looking for a particular reagent that came to Gambian airport, and I'm going to do this because it happened today. And then somebody said there were people going to Mecca. They thought that small parcel belonged to them, and this parcel is come from South Africa, stayed in Gambia customs, and now is sitting in Jeddah. So those are the challenges that we face, and I don't have any other resources to get that reagent. So I have to wait. I have to beg and see if I can get it back. If I was in London, I would just go from North London to South London, go to a lab there and collect it. So it's very difficult. So you have to plan in advance. You have to have good friends. You have to have plan B, plan C, plan D. And then you don't complain too much as well, because I think it will kill you if you keep complaining. You need to be a solution-based human being, so that every time you are thinking about a solution, not worrying about the problem, because problem will come. Oh, I mean, that's for sure.
All right, thank you so much, Dr. Karim, for that well-rounded answer. I would like to talk on behalf of the University of the Gambia. Although we know it might not be now or in the near future, but we are also hoping that we would one day become a giant in genomics research facility. So that experience really goes well to do that. So we've arrived at the end of the webinar. We see it 1 hour, and we want to stick with time. And I would like to thank you for everyone for joining this impactful session. We are truly honored to have hosted Dr. Abdul Karim Sesay, whose insights and experiences have both been enlightening and motivating. This session is a testament to what strong leadership, strategic vision, and scientific collaboration can achieve, even in the face of limited resources. Dr. Sesay's work continues to inspire many across Africa and beyond. To our audience of students, faculty, and professionals, let's take today's lesson to heart. Let's commit to growing our scientific communities and building resilient health systems with research at their core. And on behalf of the American Society for Microbiology, the Young Ambassador Program, our co-host and supporting institutions, we say thank you for being part of this dialogue, and we look forward to seeing you in our next episode. Until then, stay inspired, stay curious, and keep pushing the boundaries of microbiology for a better world. Thank you so much, Dr. Abdul Karim. It was an honor.
Thank you so much. Thank you. Such a pleasure. Thank you so much.
Thank you. And